In this video, we will discuss how proteins are made using DNA. DNA is a double-strand nucleic acid that holds the genetic code for every living organism. DNA is located inside of the nucleus, and it does not leave the nucleus. You need the information from the DNA to make the proteins. This is what your cells use to function properly. Proteins are made inside ribosomes, so the gene segment to make a specific protein needs to be copied inside the nucleus. This copy is made by the help of mRNA. Remember that RNA is also a nucleic acid, like DNA, but only has a single strand. Also, a reminder that the bases that make up DNA and RNA have one main difference. Remember, the bases of DNA are adenine, thymine, cytosine, and guanine. And remember that in RNA, thymine is replaced with the uracil. The dash represents that the two bases are paired together. So in DNA, A pairs with T, C pairs with G. In RNA, A pairs with U, C pairs with G. Let's look at an example of how protein is made. We start with a DNA strand, which uncoils so the gene segment can be copied by the mRNA. This copy of the gene segment by the mRNA is a mirror copy, which means that the copy is made up of the matching base pairs of that segment. For example, on the screen, our gene segment is AAA-CCC, so the mRNA copy is UUU-GGG. This is because A pairs with U and C pairs with G since there is no T in RNA. Refer to the base pair chart on the right of the screen and your notes to know how the bases pair to each other. Now the mRNA leaves the nucleus and enters the ribosome, where the tRNA reads the mRNA codons and makes another mirror copy. Notice that the tRNA is the same exact copy of the DNA, AAA-CCC, thus giving us the instructions to make our amino acid for that gene segment. Now let's use the codons from the tRNA to find the corresponding amino acid. Let's take the first codon, AAA, and look for it on the codon chart. The first step is to look for the first letter, which is A. You find that on the left side. It is located on the third row. Now you look for the second letter, which is also A. You find the column for the second letter by reading the top. A is found in the third column. Find the box where the third row and the third column meet, and search for the third letter, which is also A. The final step is to look for the amino acid next to AAA. The amino acid for AAA is LYS. You use this method to find any amino acid chain. Let's look at another example of this. Remember, you can pause the video and look at the examples as many times as you need to understand this. A codon is 3-base sequence of mRNA, and anticodon is a 3-base sequence of tRNA. Now we read these as codons. They are read at 3 bases at a time. Once this copy is made, the mRNA can travel out of the nucleus to the ribosome to make the protein for that specific gene. Remember, proteins are made up of amino acids. To make the protein, the message from the mRNA gets matched to the complementary bases using the tRNA. You will use the bases of tRNA strand to figure out the amino acids that will be linked together in a chain, making the protein. This part takes place in the ribosomes. The amino acid chart that we use for this part is linked as a Word document. The first step of protein synthesis is transcription. This is where the DNA gets copied into the mRNA. Once the mRNA is made, it leaves the nucleus, and this is where the second part of the process begins. The second step is called translation. This is when the mRNA codon is matched by the tRNA anticodon. You will use this tRNA strand to get the amino acids. Remember, the codon is made up of 3 bases. In order to get the amino acid, you will have to read the amino acid chart, and you do so by using this technique. The first letter or the first base is on the left-hand side of the chart, the second letter or the base is on the top, and the third letter or the base is on the right. You look for the codon and the amino acid is listed inside the box. For example, our DNA sequence is ATG, CCC, GGG, CAG. The mRNA sequence that is going to be copied from this is UAC, GGG, CCC, GUC. Remember, this is all happening inside the nucleus. Now the mRNA is going to leave the nucleus and go into the cytoplasm. So the mRNA sequence once again was UAC, GGG, CCC, GUC. The tRNA sequence is going to be AUG, CCC, GGG, CAG. All this is happening inside the cytoplasm. The tRNA sequence, which was AUG, CCC, GGG, CAG, has formed. Now we're going to use the amino acid charts to figure out the amino acids. So for AUG, the amino acid is MET, M-E-T. For CCC, it's PRO. For GGG, it's GLY. And for CAG, it's GLN. So now we have this amino acid chain and our protein is finished. Remember, this part happens inside the ribosomes. Now you have a worksheet with the data table to practice protein synthesis. Try that.